Welcome to this webinar, Advancing Protein Analysis with High-Throughput Multiplexed Fluorescence Westerns. I'm Diana Turner, Deputy Editor at DDW. In this webinar, Bio-Techne will unveil the next evolution of the LEO system, expanding the capabilities of simple Western technology for high-throughput protein analysis. This webinar will provide a first look at LEO's fluorescence performance using multiple assay models. Data highlights include precision, sensitivity, dynamic range, platform comparability, and workflow flexibility for multiplex detection. Additional datasets will showcase advanced capabilities such as resampling for up to 4 targets from a single sample, total protein normalization using near-infrared fluorescence, and RePlex Assay performance for sequential detection. Madhura Nayak is Senior Product Manager for Bio-Techne's Simple Western Solutions. Previously, she worked across the biotech and life science industry, supporting product strategy, scientific applications, and customer workflows in protein analysis, spatial biology, and cell therapy markets. Dr. Jessica Dermody is currently the Senior Director of R&D Science for Protein Simple, part of Bio-Techne. During her 15 years at the company, she has held many roles within R&D, applications, and field support. I'll now hand over to the speakers.
Thank you for the introduction. We've got a great session lined up today. I'm going to kick things off with a quick overview of Simple Western, how it works, how it's grown over the past 14 years, and then I'll introduce you to Leo, our newest addition to the Simple Western family. After that, Jessica will walk you through for the very first time Leo fluorescence performance data. She'll cover precision, sensitivity, and dynamic range. And then we'll wrap things up with an open Q&A. Before we get into the first section, I wanted to share the power of Bio-Techne with you. Bio-Techne is a global developer, manufacturer, and supplier of high-quality reagents, analytical instruments, and precision diagnostics. Bio-Techne is a powerful solutions provider with over 750,000 quality products, more than 3,000 committed employees, and over 45 years of innovation experience that can be attributed by over 500,000 citations. We also offer 7 innovative instrumentation platforms, and one of them, Simple Western, will be the focus of today's talk. Simple Western technology is fundamentally enabled by capillaries. Simple Western starts by automatically loading the separation matrices into the capillaries. This is followed by loading of a small sample plug in each capillary. Proteins are then separated by their apparent molecular weight, just like the SDS page. Proteins are then immobilized into the capillary wall using UV light. Your Simple Western instrument then removes the separation materials and performs an immunoassay inside each capillary, using the primary antibody of your choice and are ready to use secondary antibodies for easy and sensitive detection of your target. The instrument images the signal from the immunoassay in each capillary, and the data are automatically analyzed in the Compass software. Simple Western is precise and gives you fully automated results in under 3 hours. The platform is quantitative from only 3 microliters of any sample. It's incredibly reproducible, meaning you get the same great data every time with intra-assay CVs under 15%. And Simple Western delivers exceptional picogram level sensitivity with our chemiluminescence mode, and also features dual color fluorescence channels for multiplexing. Simple Western has been on the market for over 14 years and has grown to become a proven and trusted technology adopted globally with over 3,000 citations, more than 7,000 antibodies validated for Simple Western, approximately 75 pending and issued patents worldwide that protect the unique technology, and thousands of instruments installed in over 50 countries worldwide. Let me introduce you to Leo, the newest advancement in our Simple Western family. Leo brings together everything researchers love about Simple Western: automation, walkaway simplicity, and high data quality, and takes it to an entirely new level. With 96 sample throughput, Leo is designed for teams that need to generate high confidence protein quantitation data quickly and consistently. You get highly precise quantitative immunoassay performance paired with the specificity and size resolution you expect from a Western blot, all in one streamlined workflow. Another major advantage is that antibody pairs aren't required, which removes a huge barrier to assay development and gives you far more flexibility with your targets. Overall, Leo elevates lysate-based protein analysis with the precision of an immunoassay and the clarity of size resolve detection, giving you a powerful and reproducible tool for true quantitative insights. Let's take a look inside Leo to see how we've achieved this level of throughput. As I mentioned on the previous slide, Leo is built on the same proven and trusted Simple Western technology. So the workflow will feel very familiar to anyone who has used Jess or Abby before. Inside Leo holds 4 25 capillary cartridges. They look just like the cartridges you know from Jess and Abby, but these are specifically engineered for Leo. Leo also accommodates 4 new peel and go pre-filled reagent plates along with a redesigned sample plate, which streamlines setup and keeps the workflow simple and efficient. Let's walk through the key attributes that define Leo. First, Leo helps you accelerate results with a high throughput, hands-free workflow. You can run up to 100 capillaries in as little as 3 hours, making it easy to move through your experiments quickly. Next, Leo helps you save your precious samples. Each capillary requires only 3 microliters per sample or well, which is especially useful when sample availability is limited. Leo also helps you maximize data from your samples with total protein normalization using RePlex along with resample capability. So essentially, you can get more information out of every run. You also get broader multiplexing flexibility with chemiluminescence and fluorescence detection channel, which now includes NIR and IR. And Leo provides accurate, reproducible, and high precision measurements, giving you confidence in your data, and its low picogram sensitivity in chemiluminescence allows detection of low abundance targets. And finally, Leo helps you support regulatory compliance with Compass version 7, which offers 21 CFR Part 11 compliance features. With that, I'll go ahead and pass the presentation over to Jessica, who will walk us through some exciting new data on Leo fluorescence.
Thank you, Madhura, for the great introduction to Simple Western and for teeing up the discussion about Leo. I'm excited to dive into the performance highlights shown here and touch on a range of capabilities that demonstrate how Leo delivers outstanding data quality. Let's begin by taking a closer look at Leo's precision performance. Before presenting the precision results, I'll describe the experimental framework used to generate the data. To assess Leo's intra-run and inter-run precision, we used HeLa lysates and measured the signaling adapter protein 1433 and the NIR detection channel. In HSP60, a mitochondrial chaperone was measured in the IR detection channel. 24 replicates were processed under Leo's default run conditions with sample, primary antibody, and secondary antibody, each resampled from individual reagent rows as shown in the plate template in the bottom left. These 24 replicates were distributed across 4 cartridges, yielding 96 total data points. The precision study was performed on the same Leo instrument by a single operator across 3 independent runs, using identical lots of reagents, pre-filled plates, and cartridges. This controlled setup allowed for a precise evaluation of Leo's intra-run and inter-run precision across both targets and both detection channels. On the right, you can see an image showing 1 representative replicate from each of the 4 cartridges, and the peaks for both 1433 and NIR and HSP60 and IR. Here you'll see the robust and consistent performance of the Leo instrument in the qualitative lane images from the Compass for Simple Western software. We also evaluated the quantitative intra-cartridge variability to confirm the reliability of our data. CVs within each cartridge were impressively low for these models and ranged from 3.3 to 3.6% for HSP60, while 1433 inter-cartridge CVs ranged from 3.3 to 5.3%. Using these same data, we plotted the average peak areas for HSP60 and 1433 across each of the 4 cartridges, along with the run mean and the plus and minus 10% ranges from the mean. For both protein targets, the cartridge level averages fell within the 10% window, demonstrating consistent performance across the instrument's 4 cartridges. We then extended the analysis using the Leo-specific cartridge correction feature in the Compass for Simple Western software. This correction normalizes subtle cartridge-to-cartridge variation, producing a tightened clustering of cartridge peak area values around the mean. This improvement in precision using cartridge correction is also reflected in the improved intra-run CVs shown in the table on the far right. For HSP60, the intra-run percent CV improved from 6.4% without correction to 4.7% with correction. For 1433, the intra-run percent CV across cartridges was 7.7% without correction, which improved to 4.8% after applying cartridge correction. These reductions in variability clearly demonstrate the effectiveness of cartridge correction in further enhancing precision for both 1433 and HSP60 on the Leo platform. Note that within cartridge CVs are unchanged when using cartridge correction, which is to be expected. Using the same study design, we next evaluated the inter-run precision of Leo for 1433 and HSP60. We performed 3 independent runs on the same instrument using split samples along with matched lots of reagents, pre-filled plates, and cartridges. For each run, we calculated the intra-run CV both with and without cartridge correction. These results are shown on the left. As you can see, even without cartridge correction, the intra-run CVs are consistently low, ranging from 4.0% to 8.1%. With cartridge correction, the intra-run CVs are further reduced, ranging from 3.7% to 6.0%. This demonstrates the inherent precision of the Leo instrument and the effectiveness of cartridge correction in tightening performance across cartridges. On the right, the graphs display the average peak area for each run after cartridge correction, along with the overall inter-run mean and plus and minus 10% deviations from the mean. All 3 runs for both 1433 and HSP60 remained well within 10% of the mean, demonstrating consistent performance across independent runs. We also determined inter-run precision, which represents variability across the 3 independent runs. Both targets show notably low inter-run CVs with HSP60 at 4.0% and 1433 at 6.1%. These low inter-run CV values indicate that Leo delivers highly reproducible results across multiple runs, ensuring strong consistency and reliability in your experiments. You've now seen the impressive intra-run and inter-run precision on Leo, and in this study, we extended the evaluation to examine the inter-instrument precision of Leo using the same 1433 and HSP60 assay models. As before, we ran 24 replicates of the HeLa sample under default Leo run conditions, with resampling for the sample, primary antibody, and secondary antibody, generating 96 total data points per instrument. This experimental design was executed in parallel on 3 different Leo instruments by a single operator using the same lots of reagents, pre-filled plates, and cartridges across all runs. This controlled setup allowed us to accurately assess how consistently Leo performs across multiple instruments. On the right, you can see an image showing 1 representative replicate from each of the 3 instruments, and the peaks for both 1433 in NIR and HSP60 in IR. Here we demonstrate the excellent inter-instrument reproducibility of the Leo platform, underscoring its suitability for cross-site and multi-instrument method transfer. For each Leo instrument, we calculated the intra-run CVs for HSP60 and 1433 peak areas after applying cartridge correction. As shown in the table on the left, the intra-run CVs for HSP60 range from 3.9 to 5.4%. For 1433, the intra-run CVs were similarly low, ranging from 5.7 to 6.7%. These consistently low CV values highlight the strong, reproducible performance of each LEO instrument. The graphs on the right display the average peak area for each instrument along with the overall mean and the plus and minus 10% deviations from the mean. All LEO instruments fall within 10% of the overall mean, demonstrating consistent and reliable performance across instruments. When we calculated the inter-instrument precision, which represents variability across all instruments, the combined inter-instrument CV for each target was quite low at 7.0% and 7.4% for the two targets. This level of consistency shows that LEO delivers highly comparable results across multiple instruments, ensuring data reliability regardless of where the analysis is performed. Such robust inter-instrument reproducibility is essential for studies spanning multiple labs or sites, enabling seamless comparisons and ensuring that results are driven by biology, not instrument variability. Next, we will discuss sensitivity on LEO. Understanding the limit of detection, essentially the lowest amount of a target that can be reliably and accurately measured, is essential for any quantitative assay. To evaluate sensitivity on LEO, we determine the LOD for a recombinant protein, RNase, across two LEO instruments and two JES instruments for comparison. For RNase, we assess sensitivity in all three detection channels, chemiluminescence, NIR, and IR, allowing us to characterize performance across the full range of available detection modes on the instruments. For the analysis, we prepared an 8-point serial dilution in helolysate as background and analyzed split samples in parallel on both instrument types using their respective default run conditions. All samples were run in triplicate within a cartridge, and on LEO, resampling across all 4 cartridges provided 12 data points per sample per instrument. Reagents, plates, and cartridges were lot matched where appropriate to maintain consistency across all runs. The image on the right shows the resulting serial dilution series for RNase near its limit of detection for the chemiluminescence channel. Summarized on this slide are the RNase limit of detection results for the two LEO instruments. Comparing LODs across all detection modes for this protein, LEO demonstrates low hundreds of picogram to low nanogram per milliliter sensitivity, depending on the detection channel used. The low LOD values for chemiluminescence enables detection of very low abundance proteins. These results highlight LEO's strong sensitivity performance and its ability to deliver reliable LOD measurements across multiple detection channels. When we include the JES data, you can see that LEO and JES deliver comparable LOD performance across all 3 detection channels. Because LOD measurements occur close to the assay noise, it's typical for comparable instruments to fall within 2 to 3-fold of each other, which is exactly what we observe here. These results demonstrate that LEO's sensitivity closely matches JES and reinforces that both platforms can detect low abundance proteins, an essential requirement when working with critical biomarkers or therapeutic targets. Beyond LOD, an equally critical parameter for quantitative performance is the dynamic range, which we'll examine next. Using RNase as our model protein again, we evaluated the dynamic range of the LEO platform. In this study, RNase was detected across all 3 LEO detection channels, chemiluminescence, NIR, and IR. To generate the titration curve, we prepared an 8-point, 5-fold serial dilution spanning concentrations from the microgram per milliliter range down to the low picogram per milliliter range. 2 LEO instruments were run in parallel using the same lot of reagents, plates, and cartridges, and all assays were performed under default LEO run conditions to ensure consistency between instruments. The images on the right show the resulting serial dilution series for RNase in NIR on the top and IR on the bottom. The results from this study highlight the broad dynamic range for each channel, demonstrating LEO's ability to detect and accurately quantify proteins across several orders of magnitude. Here you can see LEO accurately measures RNase over a span of 3 to 4 logs, depending on the detection channel used. A wide dynamic range is essential for applications where protein abundance can vary wildly, such as biomarker discovery or other quantitative analyses. In addition, you'll notice the data points tightly cluster across the two LEO instruments for each detection channel, again highlighting LEO's high inter-instrument reproducibility. When the concentration ranges for each of the three channels are plotted on a single graph, you can see these ranges overlap significantly. This overlap is critical because it provides flexibility when analyzing targets of vastly different expression levels within the same prepared sample. For example, high abundance targets can be detected without saturating the signal, while low abundance targets remain measurable without falling below the limit of detection. By leveraging these overlapping ranges, we can confidently quantify multiple proteins in a single sample, reducing sample preparation complexity and improving efficiency in high-throughput workflows. When the data is visualized with instrument data side by side, it's clear that both instruments deliver highly comparable data. This reproducibility is critical because it means we can trust the dynamic range not just on one instrument, but across multiple systems for the three detection channels. Next, we'll take a closer look at some of LEO's additional capabilities, starting with RePlex. RePlex enables you to run two immunoassays on the same sample, which is similar to a strip-and-reprobe approach in Western blotting. The steps in a RePlex assay are outlined in the image on the right, where you can see that each sample goes through two immunoassay probing cycles, and between those probes, the RePlex buffer removes the antibodies from the first probe before introduction of the antibodies for the second probe. In the image on the bottom left, you can see the plate layout with one row of samples in orange, two rows of primary antibodies in light blue, one for each probe, and two rows of secondary antibodies in teal. For this experiment, we used resampling to run the same samples and antibodies across all 4 cartridges in a single LEO run. We detected 2 proteins in each probe, 1433 and the DNA repair protein FANCD2, using default conditions on 2 LEO instruments. For performance comparisons, we also ran a just instrument in parallel using split samples and matched reagent lots. Zooming in on the plate layout, you can see multiple probing conditions that were used to assess the performance of the RePlex assay. First, we measured the effectiveness of the RePlex buffer in removing the antibodies from probe 1. To do this, we detected any residual primary antibody from probe 1 by using the same secondary antibody in probe 2 as was used in probe 1. We also measured the equivalence between the 2 probing cycles by probing for 1433 and FANCD2 in both cycles, and then compared the immunoassay results from probe 1 versus probe 2 for each target. We'll first discuss the data for antibody removal efficiency. Effective antibody removal is essential for accurate multiplex protein analysis. Any carryover can create false positives and skew quantitation. With RePlex on LEO, we see removal performance on par with Jess. In fact, both 1433 and FANCD2, the antibody carryover to probe 2 is effectively eliminated with antibody removal efficiency of 99% or higher. You can also see this qualitatively in the lane view on the right. There's a strong band in probe 1 for each target and no band in probe 2. Confirming removal of the antibodies from the first probe before the second probe's antibodies are applied. Here we measured average peak area for 1433 in probe 1 and probe 2 on both LEO and Jess instruments. As shown in the graph, peak areas are comparable across probes within each instrument, and probe 2 values were within 7% of probe 1, confirming consistent detection across both probes. When we look at intra-run CVs for 1433, LEO delivered consistently low values for probe 1 and probe 2, ranging from 5.5% to 8.9%, demonstrating excellent intra-run reproducibility. Jess showed similar CVs, indicating that both instruments provide comparable performance for RePlex. To demonstrate how RePlex supports more traditional protein analysis applications, we detected total and phosphorylated versions of the signaling protein AKT in untreated and treated Jurkat cells. Total AKT was detected in probe 1 and phosphorylated AKT was detected in probe 2. In addition, we leveraged the overlapping dynamic ranges of the chemiluminescence channel and NIR channel to detect both targets in the same sample, since total AKT is highly abundant, while phosphorylated AKT is much lower. This study was performed across 3 LEO instruments using a single lot of reagents and split samples. Looking at the data, the lane view on the left shows clear phospho-AKT upregulation in treated cells. The electropherograms in the center highlight how closely the PAN, or total AKT, and phospho-AKT peaks overlap due to their similar molecular weights. And we can distinguish them using 2 detection channels which simultaneously confirm specificity of each immunoassay. The bar graph on the right quantifies the phospho to total AKT peak area ratio, showing about a 40-fold increase in treated samples. And this ratio is consistent across the 3 LEO instruments. This is a great example of why overlapping dynamic ranges matter. It allows you to use chemiluminescence for low abundance targets and fluorescence for high abundance targets in the same sample. Combining these capabilities provides relative quantitation through multiplexing without any additional sample prep, which saves time, increases accuracy, and provides a much richer data set. I've mentioned resampling a few times throughout this presentation, and I'd like to take an opportunity to share in more detail how this works on LEO. As shown in this video here, resampling on LEO refers to reusing the same sample or the same antibody row across multiple cartridges within a run. On LEO, you can resample a single row up to 4 times, maximizing the data obtained from a single row of sample. Here's an example of where we use resampling on LEO to detect multiple targets in each sample. We loaded a single row of samples onto the sample plate, which included 8 replicates each of HeLa lysates at 0.3, 0.2, and 0.1 milligram per milliliter, giving us a total of 24 samples. This single sample row was used to load 4 cartridges within the LEO run, and each cartridge was probed with a different primary antibody. There are 4 rows of primary antibodies shown in the plate template in the top right, 1 unique row used for each of the 4 cartridges. Like the sample, all cartridges shared the same secondary antibody, and thus there is a single row of that reagent. This setup demonstrates the power of resampling, where we can reuse the same sample and the same secondary antibody across multiple cartridges instead of preparing new lysates or secondary antibodies for each cartridge. So why does this matter? This saves time, reduces variability, and conserves precious reagents. In fact, you can save up to 50% on samples and antibodies without compromising data quality. Now let's see what resampling looks like in practice. Using one sample well as an example, we resampled that same well across the 4 cartridges in the LEO run. Each cartridge was then probed with a different primary antibody using one of the 4 primary antibody rows. The results on the left show 4 data points from the sample in well 2, representing 1 immunoassay per cartridge. So in this example, we're saving on sample volume and prep time, as well as secondary antibody volume, by using resampling on LEO. I just showed how resampling enables detection of multiple targets in a single sample. The lane view here shows the results from each cartridge for all the samples in our resampling study. But the gains in efficiency are only valuable if the data is consistent, which is what we see here. The quantitative results show low CVs for each target across the 3 sample concentrations, ranging from 3.2% to 12%. This demonstrates the excellent reproducibility and consistency in sample loading when resampling is utilized on LEO. So not only do we save on samples and antibodies, but we also maintain high precision across multiple targets and concentrations. While resampling still delivers low CVs within each cartridge, we further assess consistency of sample loading across cartridges on a per sample well basis. To do this, we calculated the peak area ratio of 3 of the targets relative to the 4th target for each individual sample. In this case, we divided the peak area for HSP27, HSP70, and HSP90 by the peak area of ERK1 on a per sample well basis. which provided peak area ratios between targets. We then normalized the ratio for each sample well to the first replicate of that sample type. A peak ratio of 1 would indicate the exact same sample quantity is loaded across cartridges. As shown in the graph on the left, all 3 HSP proteins show normalized ratios close to 1 for all concentrations. The sample loading reproducibility is further highlighted with ratio CVs well below 10%, as shown in the table on the right. As a final takeaway, resampling on Leo isn't just a convenience. It's a strategy that gives you unmatched flexibility, dramatically reduces sample and antibody consumption, and unlocks the ability to generate more data from every single sample. It's about doing more with less, while maintaining precision and consistency. I've shown you how Leo performs across a variety of assays and demonstrated key performance metrics. Madhura will now share some final thoughts on how Leo can help you maximize the value of your samples and assays.
Thank you, Jessica, for the exceptional data you shared on Leo fluorescence. This slide summarizes Leo's representative performance data, and it clearly highlights the strong reproducibility and precision that the platform delivers. Let me walk you through key points from the table. So when looking at the raw peak area CVs, both within a run and between runs, Leo achieves CVs of less than 15% across the immunoassay formats that are shown here. Additionally, we have chemiluminescence data generated across multiple instruments demonstrated in a separate study, which showed CVs of less than 10%. All this further reinforces the consistency and the reliability of data generated on Leo. Together, these results demonstrate the robustness of the platform and provide confidence in the precision and reproducibility you can expect from Leo. Let's look at how the newest capabilities on Leo can help you future-proof your research. With these enhancements, we have focused on tools that not only make your workflow faster and more efficient, but also help you generate deeper and more actionable insights. To recap, here's how. First, we've increased the workflow efficiency by expanding Leo's multiplexing capabilities. You can now extract more information from every sample, reducing the need for replicates and conserving precious material. Second, we've added new detection channels, giving you more flexibility in experimental design and enabling more complex assay setups. Third, Leo now supports multiplexing targets with different expression levels within the same sample, which is especially valuable when analyzing complex biological systems. Together, these updates open up new possibilities for your experiments. For example, you can now measure phosphorylated and total forms of a protein in the same capillary, giving you deeper insight into protein modifications with far less time and effort. And with expanded multiplexing levels, you can begin to map out more components of a signaling pathway in a single run, helping you see a more complete picture of your cellular processes without the need for multiple time-intensive experiments. And with that, I would like to thank you all for attending today's webinar. We focused on Leo fluorescence, and we hope the data we shared was helpful as you explore what Leo can bring to your research. If you would like to learn more about additional Leo capabilities, we've included some resources on this slide that you can access after the session. And now we're ready to move into the Q&A, and we'd be happy to answer any questions you may have.
Thank you very much for those excellent presentations. If you're ready, we'll now get on to the Q&A. Okay, brilliant. Okay, so our first question is for Jessica. So the question is, I want to make sure I'm understanding resampling. Does this mean we can set up a single 24-well cartridge and then supply 4 sets of capillaries using that one cartridge? Does that mean we can buy the cartridges and the capillaries separately?
Yeah, so thanks for the question. Just to clarify, the cartridges are provided as a set of 25 capillaries. So you can't actually treat each individual capillary separately. What we're talking about with resampling is essentially taking that 25 capillary cartridge and then loading either multiple rows or a single row of sample multiple times, or a single row of antibody multiple times across different cartridges. And again, each cartridge has 25 capillaries. So you can't actually have the capillaries separate from the cartridge. They're kind of one and the same.
Okay, brilliant. Thank you very much. Coming over to you, Medora. Our next question. Can I run 1, 2, 3, or 4 cartridges in a Leo run, or do I always need to run all 4?
Yeah, thank you for that. As we mentioned, Leo is sort of built for flexibility. So yes, you can run any number of cartridges, 1, 2, 3, or 4. And as Jessica mentioned earlier, each cartridge has 25 capillaries. But what happens is with each cartridge, you also require its own pre-filled reagent plate. And every run will require a fresh sample plate. So running fewer than 4 cartridges simply means that you'll use fewer reagent plates, but you still will need a dedicated sample plate for every run.
Perfect, thank you. Coming back to you, Jessica. How long does it take for Leo to analyze 96 samples?
Yeah, so it actually depends on what type of assay you're running. So, we showed in this presentation today, Leo's capable of 3 different detection channels, and that's really kind of the driving force for how long the run will be. So if you're running a standard chemiluminescence assay, you can actually get data for 96 samples in as quickly as 3 hours. If you start combining fluorescence channels into those runs, where if you're doing multiplexing, can range from anywhere about 3.5 to 5 hours. Again, it just depends on the combination of detection channels you're using. Another wrinkle to that is if you're running a RePlex assay. So again, a RePlex assay is 2 immunoassays per cartridge. So those run times tend to be 5 hours if you're only using chemiluminescence, but can be a little bit longer as you add more detection channels into the run. So shortest 3 hours for chemiluminescence, and then 5 hours or a little bit longer if you're adding multiple detection channels or running RePlex.
Okay, great. Coming back to you, Medora. I have an existing Jess. Are Jess and Leo consumables interchangeable?
That's a great question. And well, as we mentioned in the webinar, right? So the plates and the cartridges are instrument specific. So Leo has a redesigned new sample plate, and the cartridges are also specific to the instrument. However, some of the reagents, such as for Simple Western, if you're familiar, you use the separation modules and you use detection modules. So components of that, some of the reagents can be shared, the detection modules can be shared, but there are instrument-specific plates and cartridges that need to be run with the Leo.
Okay, thank you. And I think this is one for you, Jessica. Can I run different assays on each cartridge within a single run?
Yeah, so it's a little nuanced, but essentially all the cartridges must have the same run conditions. So things like separation and incubation times. However, you can run different immunoassays. So different sample types, different antibodies across the various cartridges within a run. So, given this, you have some flexibility on the assay setup, especially around the samples and antibodies, and you can combine that with resampling or RePlex and really increase the number of essentially immunoassay data points that you get. So while we can run lots of different immunoassays, just the run conditions have to be the same across the cartridges.
Brilliant, thank you very much. Next question is, what are the onboarding workflow requirements for Leo with reference to sample prep and reagent loading? And that's for you, Medora.
Yeah, so as you mentioned, like Leo basically uses the capillary cartridges. There's pre-filled reagent plates that reduces the setup time, and then there's a dedicated sample plate. So as users, you would add samples, your primary antibodies, your secondary antibodies, some additional reagents into the sample plate, like wash buffer. You use luminol peroxide for detection, for chemiluminescence. So all of that gets added to the sample plate. And that's essentially how quick it is to sort of load a Leo run and set that up.
Thanks very much. Coming back to you, Jessica. I use a Jess instrument and can perform total protein normalization in my assay. Can Leo do the same?
Yes, so we didn't talk about total protein normalization on Leo. We did have a webinar earlier last year about the chemiluminescence assays on Leo. And so we do talk about total protein normalization during that webinar and give some example data. But generally speaking, yes, Leo can run all the assays that Jess can. Really what's new on Leo around total protein normalization is that you can either do normalization within a cartridge like you do on Jess today, or if you have multiple cartridges where you're running total protein normalization, you can normalize within the cartridge or normalize across cartridges. So again, this kind of gives you that flexibility of running more samples and allowing you to compare data across cartridges within a Leo run.
Excellent, thank you very much. Our next question for Medora. Can you speak a little more about the software and does it support regulated or QC workflows?
That is a great question. So Simple Western users might be familiar with Compass, and so Leo actually also runs on Compass for Simple Western. And essentially that is the software that users would use to set up the assay. It also has some new data analysis capabilities. It also has some reporting capabilities to make that reporting straightforward as possible. So with the Compass software, there are some new built-in tools. As you saw, some great data that was generated by Jessica. There's some standard curve generation tools. There's quantitative concentration reporting tools. You can group samples given that you're running so many samples on the Leo. And another one that Jessica also showed in all of her data was with cartridge normalization. So that sort of like helps in reducing some of the manual data handling, and it also helps improve sort of like workflow consistency. And to address the question, yes, Compass supports 21 CFR Part 11 compliance. And what that means is it does have features or supports features like secure user access and audit trails. So that is what makes that suitable for regulated and QC workflows, where data integrity and traceability of that is important.
Brilliant, thank you. Coming back to you, Jessica, now. If we use resampling with RePlex, how many different antibodies can be used to probe a single sample?
Yes, so with resampling, let's assume you're going to have a single sample row and you're going to resample that across the 4 cartridges in the Leo run. So if you combined this with RePlex, you could actually have up to 8 unique antibody rows. And so it's 4 antibodies for probe 1, and then it's 1 antibody per cartridge. And then when you RePlex and do that probe 2 on each of the cartridges, you'll have an additional 4 antibody rows that you can use. So again, it's 4 cartridges times 2 immunoassays per cartridge, so you can get up to 8 antibodies.
Excellent, thank you. And Medora, now this one's for you. What types of samples work with the Leo system?
Yeah, so I think following up on the samples that Jessica showed in some of the data, we do use Leo can work with a wide range of sample types, right? So we understand that researchers are using different types, so it can work with like cell and tissue lysates, purified proteins, more complex biological samples. And one of the things that we also touched upon in the webinar was that it only requires about 3 microliters of the sample. And with just that much amount, you can see the, it can give like highly sensitive data. And it's a great option when, sample amounts are very limited. And also like, when you're working with like low abundance targets, you can use, a low amount of sample as well. And in addition to that, like, primary cells or, valuable tissue samples, all of that works with the Leo. So it does support a very wide range of sample types.
Perfect, thank you. And our last question is also for you. I have an existing Western blot assay. Will I need to revalidate my antibodies when moving from traditional Western blots to Leo?
Yeah, that is a great question, and we do get that often. So in many cases, we have observed that antibodies that work well in a traditional Western blot will also perform on the Leo. However, some optimization is usually recommended. So again, it's on a case-by-case basis. General observation is most likely it'll work, but there'll be some optimization that will be needed. And we actually have a really great support team that can help, guide you through some of the optimizations, which could be with respect to antibody dilutions or, incubation conditions, and some other assay setup features, so that we can ensure that, you're getting reliable performance with Leo or any of the other Simple Western systems. So.
Brilliant, thank you very much. I think that's all our questions for today. So thank you so much for those detailed responses and for your time. And thank you to our audience for joining us. If you'd like to watch again, this webinar will be available on demand at ddw-online.com, and you can access Drug Discovery World's news, features, podcasts and webinars on your mobile and tablet wherever you are.